everyone, and thank you so much for your presence. Today, I will be defending my dissertation where we engineer E. coli bacterial strain for the production of indirubin derivatives using cytochrome P450 enzyme that was identified in the plant Isatis tinctoria, which is also commonly known as woad. I will be talking about what is cytochrome P450, what are its roles, what is indirubin, its limitations, and why we are interested in indirubin derivatives. Then my objectives, and then I will be discussing about my results for the production of indirubin derivatives, its biological activities, enzyme purification, and a preliminary chemical characterization of the compounds. Cytochrome P450. It's a heme monoxygenase enzyme that has been identified in all kingdoms of life. Heme meaning, as shown in this figure, this enzyme has a heme domain. And this heme domain is a part of its active site, which also has, and this heme domain has iron. And it's called a monoxygenase enzyme because once this substrate binds to the heme domain of the enzyme, this substrate adds one oxygen atom to the substrate from the molecular oxygen, producing this hydroxylated product. And for this reaction, NADPH is also required because it supplies electrons, which is supplied by this P450 reductase enzyme. So today we will be discussing about the P450 that is present in plants. In plants, the P450 and the P450 reductase is found in the endoplasmic reticulum. And plus, in this figure, you can see how the electron is being transferred from the NADPH by the reductase to the P450 enzyme for the hydroxylation reaction. And in plants, these hydroxylation reactions are a part of different types of metabolic pathways for the production of bioactive compounds. For example, vinblastine, vincristine. These are anti-cancer compounds found in the plant Madagascar periodicum. Also, artemisinin, it's an anti-malarial drug that is found in the plant sweet wormwood. And the bioactive compound we are interested in is this red pink colored compound known as indirubin. And this slide shows the pathway plus the P450s that are involved for the production of this compound. And for now, only one type of the P450, that is the CYP71B102, has been identified in this plant, Isatis tinctoria, that takes part in the production of this indirubin compounds. And also, when the extract from this plant is collected, the extract has more of this blue-colored compound known as indigo instead of indirubin. So, the first limiting factor of indirubin is its yield because it is only produced as a side product. The main product is indigo. But still, we are interested in indirubin. Why? Because indirubin has different biological activities. And indirubin is mainly studied for its anti-cancer property. And it was also identified as a main ingredient in the traditional Chinese medicine that was used to treat chronic myelolytic leukemia. And indirubin has all these different types of biological activities because it can inhibit different cell cycle kinases like CDK or cyclin-dependent kinases, which I will be talking later. But another limitation of indirubin is its water solubility. It is not soluble in water. So we know that the first limiting factor of indirubin is its low yield, which leads to inconsistent supply and obviously high cost. Then comes low solubility. It's not soluble in water. So that's why it leads to low cellular activity and poor absorption. So that's why all these limitations limits the pharmaceutical application of indirubin. So instead of indirubin, different types of indirubin derivatives have been produced by attaching functional group in the side chain of indirubin. But all these different types of indirubin derivatives that have been produced are produced or synthesized chemically using hazardous and expensive chemicals. That's very toxic to human health plus to the environment as well. So that's why we are proposing an alternative method for the synthesis of indirubin derivatives using microbes. In particular, using an E. coli strain that has been engineered with the plant P450 gene that was identified in Isatis tinctoria, along with the plant P450 reductase. And this reductase is found in Arabidopsis thaliana plant. Why? Because microbes can produce the precaution molecules. The main precaution molecule that is required for the production of indirubin derivatives are tryptophan and indole. And both of these precaution molecules are produced in bacteria. And in fact, heterologous expression of this P450, the plant P450 and the plant reductase, when it was done in E. coli by Lakshmi, she is one of the students from Dr. Antarola's lab, when she expressed that gene in E. coli and cultured that engineered E. coli strain in half TB medium, she found blue colored bubbles forming on the surface of the culture. And she also confirmed that the blue extract has 3 of the indigoid compounds. That is indirubin, isatis, and indigo. And obviously, the high was indigo. So this also confirmed that the plant cytochrome P450, that means the CYP71B102 that was identified in Isatis tinctoria, is able to oxidize indole for the production of indigoid compounds. So what we did, we used the same engineered bacteria E. coli strain. And we cultured this E. coli strain in LP medium. We supplemented IPTG, aminolevulinic acid, thymine, and tryptophan. Tryptophan is the main precaution molecule for the production of indigoid compounds. So what we were expecting more of blue color because that's the main product that's supposed to be formed. But to our surprise, instead of blue, the whole culture turned pink. So this is the color of indirubin. So what we did was we used the same system now to test whether this same system can produce indirubin derivatives, for which these are my specific objectives. First is engineer the E. coli strain for the production of indirubin derivatives. So for engineering the bacteria, first we engineered the bacteria with the plant P450 gene along with the plant P450 reductase. Also, we engineered the bacteria with the codon optimized P450 gene and the codon optimized P450 reductase. But first, the engineered E. coli, which has the plant P450 and the plant P450 reductase. This is what the culture looked like after 72 hours of incubation. We then centrifuged the whole culture for pelleting the cells, and this is what the pellet looked like after we removed the culture. Then we added water just for washing the cells, and then we found accidentally that whatever the compound is here that is pink is easily soluble in water. So this extract, this is the water soluble extract. Pink, color like indirubin, but unlike indirubin, it's soluble in water. So this was our first indication that maybe something is being produced over here that is a derivative of indirubin. We named this extract as LJSA023 extract, green in color. Green color, the extract that is harvested from the engineered E. coli, which has the plant P450 and the plant P450 reductase. Then, when we were doing the shape class, that means when we were culturing the engineered strain, we also performed spot testing. For this, what we did was we just evaluated the cells at different time points, diluted and did a spot testing. Here you can see for the engineered strain, the strains were only able to grow up to 24 hours at different concentration compared to the engineered strain in case of the control. Control meaning the cell that the E. coli cell that's not transformed or it's not being engineered. What we found, it's able to grow at different time points, even from 24 to 72 hours at different concentration. So this means that when we are culturing this engineered E. coli strain, something bioactive compound might be producing, which might be inhibiting the growth of the cell. Then, what we did was we thought that maybe the cells are being inhibited because maybe that whatever is being produced might be damaging the cell. For that, we performed dead cell testing using this propylium iodide dye. It's a fluorescent dye. How this works is that if we have a healthy cell, the cell membrane is intact. When we add propylium iodide, it stays outside of the cell and the fluorescent intensity is very low. But if we have a damaged cell which has this cell membrane damaged and we add propylium iodide, the propylium iodide will penetrate and go inside of the cell and bind to the DNA molecules. Once it is bound, this will give a very high fluorescence intensity signal. Higher the signal, potentially because the propylium iodide is binding to the DNA. Lower signal, that means potentially the propylium iodide is outside of the membrane. So for this assay, when we were culturing the engineered E. coli cell, similarly, we evaluated the cells at two different time points and then added the propylium iodide dye and measured the absorbance. For the control, here you can see the signal is very, very low at both the time points. Whereas for the engineered strain, the signal is very high. Compared to time zero at 72, after 72 hours, when we perform this assay, the intensity is even higher, indicating that maybe this is because of membrane damage. It supports our earlier result as well. Now, after this point, what we were doing, expressing a eukaryotic gene in a prokaryotic organism. So what we thought was, okay, we will do a codon optimization of the eukaryotic gene and then express it in the E. coli. Now, why codon optimization? Because whenever we express a eukaryotic gene in a prokaryote, the protein of interest that we want is very low in concentration. It's because the amount of tRNA is very low. tRNA that is required for reading this codon that codes for the respective amino acid. If we do a codon optimization and then express it, the protein will be very high because the tRNA is available. Then the rate of translation will also increase, producing more of the protein of interest. So we did a codon optimization and then engineered the E. coli strain with the codon optimized P450 and the P450 reductase. This is what the culture looked like after 72 hours. There was the pigment development and plus the pigment was water soluble. So this is the water soluble extract. And this we named it as LJSA236 extract, which is pink in color. P meaning the extract that is harvested from the engineered E. coli, which has the codon optimized P450 and the codon optimized P450 reductase. So what we found was we were able to engineer the bacteria, and we were also able to harvest or isolate the water soluble extract. And we also found that during the production or during the shake flask experiments, the cells, the engineered cells are being inhibited and potentially maybe because of damage. Now, moving from the first to the second objective, we try to characterize the biological activities to know whether the extract also has some biological activities like that of indirubin or not. So for the biological activities, first we tested the extract, the 023 extract against prokaryotes and eukaryotes. In case of prokaryotic organism, we tested it against this Salmonella typhimurium. This is a pathogenic strain. What we did was, so here these are all the different concentrations of the Salmonella. And then we tested it against the 023 extract, 0, 25, 50 and 100 microliters. Compared to 25 and 50 per 100, what we noticed was when the concentration of the Salmonella was at 10 to the power minus 4 and 10 to the power minus 5, you can see at this concentration, when it is treated with 100 microliters of the extract, we have lesser number of colonies compared to that of the control where the extract was not added. This means maybe the extract has some compounds that has bioactive properties that is inhibiting the growth of the bacteria. So maybe the extract is showing some potential antibacterial activity. Then after that, we tested the extract against HeLa cells. HeLa cells are cancerous cells. So for this one, after the treatment, we counted the number of HeLa cells after the treatment at different time points. So here you can see for the control, the number of cells, the cancer cells are increasing with time. As we know, cancer cells are highly proliferative, so it's increasing with time in case of the control extract. But when the HeLa cells were treated with the 023 extract, the number of cells were decreasing over time, indicating maybe the extract has some bioactive compounds that is showing this anti-cancer property because it is inhibiting the growth of the cancer cell. Then we tested it against yeast. Specifically, we used this yeast strain, Saccharomyces cerevisiae BY4703. Why is? Because it's a model organism for carrots. So, different concentration of yeast cells and different concentration of the extract. At 10 to the power minus 3 concentration of the yeast cells, we noticed the growth was inhibited when it was treated with 200 and 500 microliters of extract. So this also supported that maybe the compound that is being produced is bioactive and has antifungal properties. Now moving on, from the 023 extract, we tested the biological activity of the 236 extract. For this extract, we tested the biological activity against different yeast strains. What we did was we treated the yeast with the extract, 236 extract, and then we measured the optical density, OD 600 at different time points. And then we monitored the lag phase and the growth rate. So lag phase is the initial time the yeast cells or the cells take before they start to grow. Whereas growth rate is the rate at which the cells are dividing at a constant rate. So why we did measure this lag phase and growth rate was because when the cells were treated with this 236 extract, we noticed that the cells were taking more time to grow. The lag phase was extended. In addition to that, what we found was the growth rate being reduced. So we measured lag phase and growth rate. Growth rate we measured using this formula. This is showing the growth rate and the lag phase when the yeast Saccharomyces cerevisiae being treated with 236 extract. So for the growth rate, at different concentrations of the yeast cells compared to that of the control, you can see the growth rate being decreased when the yeast cells are being treated with the 236 extract. Simultaneously, for these three concentrations of the yeast cells, you can see that the yeast cells have an extended lag phase. That means the cells, when being treated with the extract, are taking more time to divide. Then we tested the extract against pathogenic yeast cells as well. So for the pathogenic, we picked two pathogenic yeast cells, Candida albicans and Cryptococcus neoformans. So same, we treated the pathogenic yeast cells, measured the growth rate and the lag phase. So this is for Candida. When the different concentrations of Candida cells were treated with the control versus the extract, 236 extract, the growth rate is decreasing. At the same time, the lag phase is increasing. Same similar result for the other pathogenic yeast cell as well. So this also supported our data that when not only the normal yeast cells, but when the pathogenic yeast cells are also being treated with the 236 extract, it is the pathogenic yeast cells are taking more time to divide. Now, up to this point, we know that the extract that we isolated or harvested has some biological activity. It can inhibit the growth of prokaryotic cells, and it can also inhibit the growth of eukaryotic cells. And particularly when this 236 extract is being treated against the yeast cells, the cells are taking more time to divide. Now, to have an idea about what are the genes that the extract is affecting, because of which the yeast are taking more time to divide, to study that we screen yeast mutants. For screening the yeast mutants, we picked this Saccharomyces cerevisiae BY4743 yeast mutants. Now, we all know cell cycle process is highly regulated. And for a yeast cell, if a yeast cell has to go from G1 to S phase, the first cycling that needs to be activated is CLN3 cycling. This is the first cycling that needs to be activated in yeast for a cell to go from G1 to S phase. So always, cycling cannot perform by itself. Cycling needs to have its partner, that is cycline-dependent kinase. Once this CLN3 cycling binds with its partner, this kinase enzyme phosphorylates this inhibitor, releasing transcription factors. And then this transcription factors further activates downstream genes, because of which the cell can go from G1 to S phase. Along with the cycline and cycline-dependent kinases, there are other cycline and cycline-dependent kinases, activators, and inhibitors that regulate this cell cycle process. And cycline and cycline-dependent kinase not only regulate this cell cycle process, but also regulates another process, which is the transcription process. During the transcription, mRNA is formed. So when an mRNA is formed, we have initiation, elongation, termination, and recycling. And all these steps are highly regulated. Phosphorylates the RNA polymerase II enzyme for regulating this transcription process. And remember this CLN3, this is the first cycline that needs to be activated in these cells for the cell to go from G1 to S phase. And the activation signal for CLN3 also comes from this transcription process. So that's why we selected few of these mutants. Mutants meaning the genes, these particular genes that regulates the cell cycle or this transcription process, the genes, those genes in that mutant is deleted. So here's the list of the yeast mutants. These are the BY4743 mutants that we selected. So we treated this yeast mutant with the extract control and the 236 extract. measure the tolerance index. What we found for these 3 of these mutants, we noticed that when the mutants were treated with the 236 extract, it has a higher tolerance index compared to that of the control. That means even though these mutants have these gene deleted, the treatment that we are giving, it is able to tolerate the treatment that we are giving. And we found out that all these 3 of the mutants are the genes that are responsible in regulating the process of transcription. Then remaining for the 5 of the mutants, we found that the tolerance index is low compared to that of the control. Since the gene is being mutated, since these genes are mutated in this mutant strains, those strains are sensitive to the treatment that we are providing. And we also found that this remaining 5 of the genes or mutants have the genes that regulate the process of cell cycle. So maybe, potentially, maybe that these cells are taking more time to divide when we are treating it. It might be because the cells are being arrested in G1 phase. Next, we measure the concentration of endotoxin. Now, why endotoxin and what it is endotoxin is also known as glycopolysaccharide that is found in the outer membrane of the E. coli cell. It's found not only E. coli, in all gram-negative bacteria. It has 3 components: O antigen, oligosaccharide, and PPA. And this endotoxin, if a bacteria is disrupted, it gets easily released. And plus, it has biological activity as well. So to confirm that the extract that we have and the inhibition effect that the extract is showing is all because of the compound that is being produced rather than endotoxin. Because the extract that we are using is a crude extract that is being isolated from the gram-negative bacteria. So we measured the concentration. As expected, endotoxin is present in the extract. But even though endotoxin is there, the extract is showing effect. The yeast cells are taking more time to divide. That means this effect is because of the compound that is being produced rather than the endotoxin that is there. Now, from this, what we learned was that the extract that we have has biological activities. It has different biological activities. Potentially, it showed like antibacterial, anti-cancer, and antifungal activities. And it also might be arresting the cells at G1 phase. Now, from the second to the third objective, where we try to purify enzymes. So, for the enzyme purification, first we try to purify the plant cytochrome P450, then the codon optimized P450, also the codon optimized reductase, and a chimeric protein. For the purification work, we attached a 6X histidine at the C terminal of the gene and regulated this with the T7 promoter. For the purification, also we used E. coli, we used BL21, and the purification we used chemical columns. So for the plant P450 gene, we expressed it at 37 degrees Celsius for 5 hours, and then we harvested the total protein, and then we ran the gel and the Western. The expected band size is 55.5 rad. So this band over here is from the expression of the P450. Then we tried to purify the proteins. Even we tried other different conditions, but we were not able to purify. So what we did was we tried with the codon optimized P450. So for the codon optimized P450, we tried it at 20 degrees Celsius for 48 hours. So same, we expressed the gene at 20 degrees Celsius for 48 hours, isolated the total protein, and this time we tried to purify it. And then during the purification, we did the gel run and followed by it. This is the Western. So the expected band size for this was also 55.5. And this was the one that confirmed that we have the purified codon optimized P450. Then, since we had the purified codon optimized P450, we tried for the codon optimized reductase ATR2. For this one also, we expressed this gene at 20 degrees Celsius for 48 hours, and we isolated the total protein and did the gel. So this is what we got. So we observed that the ATR2 protein is being fragmented and a lot of protein is being lost. This is the band size, that is 62, which we are losing the protein. And the expected band size for the codon optimized ATR2 is 73.63 kilodalton. Then we constructed a chimeric enzyme. Chimeric enzyme, by linking the codon optimized P450 with the codon optimized reductase. As a linker, we used a rigid linker because this linker has been used for other monoxidase enzymes. And the expected band size for this one was 130.46 kilodalton. And we tried the condition at 20 degrees Celsius for 48 hours. So this band over here confirmed the expression of the protein, but we were having the same problem of fragmentation. And this fragmentation pattern looked similar to that of the fragmentation pattern of ATR. We were able to purify the codon optimized P450, and we were only able to express the chimeric protein. Now moving from the third to the fourth specific objective. So for the fourth one, we did a preliminary characterization of the extract to identify the compound that is present. For this, we did different analytical techniques, and mainly we looked at the spectral property of the compounds. So first is the UV visible spectra analysis. UV visible spectra analysis helps to identify the compound based on its maximum absorption wavelength, so the maximum wavelength. And for indirubin, we know that the maximum absorption wavelength is at 540 nanometer. Yes, indirubin has other peaks as well, but the characteristic peak of indirubin based on literature is 540 nanometer when we go visible. And when we did a UV visible for the extract, this is for the extract, the brown color spectra is for the extract, we got a maximum absorbance at 408 nanometer, plus 2 tiny peaks at around 550 nanometer. So comparing these two peaks, we could conclude that the compound that is here in the extract is different than that of indirubin. It is not indirubin, but something different. Then we did a Raman. Raman Raman gives us idea about the functional groups that is present in the extract. Functional group that is present in the molecule. And for indirubin, this is the brown color peaks are for indirubin. So these peaks are the functional groups that is present in indirubin, and it is known. And this is like a fingerprint for indirubin. These peaks are for this functional group which appears at this particular wave number. When we did the Raman for our extract, the blue one, this is our peak. So what we found was that when we compare the peak for indirubin and for the extract, some of the peaks were overlapping. That means the functional group that is present in indirubin might also be present in the compound that is here. In addition to that peak, there were some distinct peaks as well. These peaks are yet to be characterized. What are the functional groups? So this also supported that the compound that is in the extract is not indirubin, but it might be something similar to that of indirubin. Then last, what we did was tandem mass spectrometry. This is a very highly specific and sensitive technique. So how this works is we have our samples, we ionize the samples and then generate molecular ions. We have lots of molecular ions which have specific mass by charge ratio. Based on this mass by charge ratio, it is identified. Then for the standard mass spectrometry means only one molecular ion is picked and then it is further fragmented. Each of the fragment gives, it's fragmented producing product ions. Each of the ions has specific mass by charge ratio. Based on this fragmentation pattern and this mass by charge ratio, the molecular structure of the compound is determined. First, we did the LC-MS of indirubin. LC-MS is also known as the tandem mass spectrometry. So, for the LC-MS of indirubin, we picked the method that is from our literature and then performed the LC-MS. This is the fragmentation pattern for indirubin that we got. When we compared the fragmentation pattern that we got with the article, it was same because it had these characteristic peaks. So the method was confirmed, so we moved forward with the method. Then when we did the LC-MS for our extract, so remember for the tandem mass, for the LC-MS, first we have the molecular ions, then we pick one molecular ion and then we get product ions. In our case, we pick 3 molecular ions. Those were abundant. This is the fragmentation pattern for the first molecular ion, which had the mass by charge ratio of 261. And based on this fragmentation pattern, this is the potential structure that is predicted. Comparing this with indirubin, you can see 2NH being added or replaced in the 3' and 3 position of indirubin. The second is, this is the fragmentation pattern for the second molecular ion, which had the mass by charge ratio of 262. Based on this pattern, this is the molecular structure, potential structure predicted. NH is the functional group being substituted in 3' when it is compared to indirubin. Then, this is the fragmentation pattern for the abundant molecular ion, which had the mass by charge ratio of 377. And this is the functional group that's been added in the 6th position when you compare it to the indirubin. So, we were, based on this LC-MS data, we were able to predict 3 potential structures that might be present in the compounds that might be present in the extract. So, for this objective, the take-home message is we were able to predict 3 potential structures of the compounds, and when the structures were compared with that of the indirubin, we were able to say that maybe it is indirubin derivative that is present. So I would like to summarize saying we engineered the bacterial strain with the P450 gene and the P450 reductase gene for the production of water-soluble extract, which also had different biological activities. And similar to that of indirubin, it also showed a potential anti-cancer property. There are lots still that needs to be done, like for example, purifying the kinase protein. After the protein is there, then we can further do the biochemical characterization of the gene. Then we need to confirm the structures, the molecular structures of the compound that is present in the extract and doing genome editing. Because all the engineering that I did so far was plasmid based, and compared to plasmid based, doing the genome editing is more stable. So I would like to thank my advisor, Dr. Angela, my co-advisor, Dr. Jaikodi, all my committee members, Lakshmi, Dr. Shiva, he helped us with the analytical techniques, all my lab mates, and Besmir and Pitur, they helped me with the biological activity. Dr. Fisher and Dr. Jose for providing me with the strains, and for funding, I got a doctoral fellowship, and I was a teaching assistant before that. SI Bridges, they helped me during summer, and the CALPS and OBCR standard one. Thank you.